Welcome everybody to the Not So Serious Science Podcast, a podcast that we talk science so that your grandparents can understand. I'm your humble host, Pat, and my partner in crime is Emmanuel.
Hi everyone. So today we have a special guest that's going to join us for our podcast. He basically integrates material science, drug delivery, and pharmacokinetics to develop models to translate nanomedicine to the clinic. Now that was a mouthful for myself, so I'm hoping our guest will be able to explain that a little bit better than what I just did. So it is my pleasure to introduce Dr. Joshua Reineke, a professor at South Dakota State University. Welcome, Josh. How are you today?
Great, yeah. Thanks, Emmanuel and Pat, for having me on today. And you set up a high bar there. So
we're super excited to have you on our podcast today. So thank you and for taking the time to chat with us. Can you tell us a little bit about what you're working on right now?
Well, right now we're working in two disease areas: pancreatic cancer and tuberculosis. And so we're just continuing that work. It's obviously, as I'm sure it is for a lot of people, slowed down a bit with the restrictions to building access and so forth.
So when you're working on TB, what do you mean by working on it? What are you developing, or what is this product that you're potentially working towards?
Yeah, so that project I think is really exciting. My graduate student Eileen Guo has really worked hard on this project. But what we're doing are developing biomimetic nanoparticles, or actually microparticles, that can get into the macrophages and that where the tuberculosis resides. And since they are mimicking the tuberculosis, we hope that they get inside the same cells, behave similarly, traffic similarly, and then the idea is to then activate the macrophage so that it is effective against both the tuberculosis infection, but also against multi-drug resistance. And tuberculosis has a problem where it can be latent, so hopefully this will be active against latent disease and looking towards eradicating disease that way.
So when you say biomimetic, well, what does that mean exactly, to mimic the virus? Is it like a little miniaturized robot, or how is it going to mimic the virus? I'm kind of curious to kind of learn more about that.
Yeah, so really it's a lot more simple than that, than like a robot. We're matching the shape and size of the bacterium. So tuberculosis is about 2 microns in length. It's kind of a rod shaped and about 200 nanometers in width. So these rod shaped shapes of the bacteria we mimic by using a specialized kind of particle which can grow in rods. So they grow as crystals. These are metal organic frameworks. But we grow these frameworks, these porous frameworks, in that same size. So we're able to mimic the shape and size of the bacteria very specifically. And then on top of that, we can take the coating of the bacteria or key coating materials of that bacteria and conjugate them on the surface of these particles too. So from a cell's point of view, it will look very similar or the same as the bacterium.
So the cell is essentially fooled. Is that like it just thinks that it's TB and reacts to it exactly the same way?
Yeah, that's the idea. So tuberculosis has been very clever over years of development and has found ways to interact very efficiently with macrophages and turn them into a host cell where they can live and proliferate within that cell. And so we want to be able just to hijack those same mechanisms that they've developed into getting in the same space.
So after these biomimetic particles get taken up into these immune cells, right, macrophages, what happens next? Is it still going to be able to recognize the actual tuberculosis, or is it kind of acting like almost like a vaccine in that sense?
Well, so here's where it moves into the theoretical and what we're proposing in the current work that we're doing. So we just got an NIH grant, and I should mention my collaborators. So Admir Dubé at University of Western Cape in South Africa, and Samantha Sampson, who's at Stellenbosch University, also in South Africa. What we're doing are taking these particles in various tuberculosis models and seeing exactly how the macrophage responds to them. The idea and the theory behind it is that we can activate these macrophages such that they then clear the infection. That is done a little bit just by the nature of the surface coating of these particles, but we can also look at administering therapeutics through these particles too that could have that same action.
Oh, very cool, very cool. So how prevalent is tuberculosis in, for example, North America compared to developing countries? Is it much of a concern here nowadays in the U.S. or rest of North America?
Yeah, that's a great question. I think something that's a little surprising to a lot of people, and that it really is quite prevalent. You can take the general population just about anywhere in the world, and 1 in 3 individuals will be infected with tuberculosis. The major difference from developing worlds, developing nations, and their higher rate of tuberculosis disease is mostly due to different stress factors or stressor factors in the life that then allow that disease to develop from the infection. But it's definitely something that's of prevalence here in the U.S. as well. We just had an outbreak at a campus that's about, I guess it's about 50 miles from where we're at in Minnesota that had a tuberculosis outbreak where 30 individuals, 30 students contracted tuberculosis. So it is still a problem in the U.S. Yeah.
Wow. Yeah, you don't really hear that much about that, but it's just it's amazing how it's still prevalent like that. Wow.
Yeah, and I think the other factor to that, it is a treatable infection. So in most cases it can be treated, but the treatment is very complex. It's usually a 6 to 9 month treatment regimen of at least 3 antibiotic drugs. And so in a developing world, that's a little bit easier where there's greater access to healthcare providers and so forth. But in an underdeveloped nation, then that becomes a little bit of a barrier in that you don't have as great of compliance to that treatment.
So these are basically nano-sized particles, right? So what is nanomedicine and nanoparticles per se? Just for our audience, to kind of understand, if you can explain a little bit. I mean, to kind of, how do you put that into perspective? Like, can we see this with our naked eyes, or?
Yeah, and I think so just a quick clarification on the nano. So in this project, we're using micro. We are doing some nano approaches as well, but I think sometimes the hype of nano, we shouldn't get too carried away with it, and that there's some great abilities of micro things. So things on a higher size scale, like this mimicking the size and shape. The bacteria are on the micro scale. But we are testing nanoparticles too, because we may be able to provide more drug or get more into the cells using a nano approach. So we are taking that. So I think nano is I guess the best way to put it are just utilizing very, I guess, unique phenomena that are on the small scale of things that are in our body, like proteins and so forth, so that we can utilize the chemistry, the surface tension, the surface area to really take advantage of some biological properties, because these are the same type of scale that a lot of the biological entities that work our body are working on, is on that same scale. So,
our podcast is really about explaining science to people like our grandparents, right? People like our neighbors, to really understand science. So I think you developed a nice tool or a nice technique to explain this. Can you tell us a little bit about that?
So I was a big fan of, the Bob Barker era, Price is Right. So I really love the game Plinko. I was one of those kids who got all excited. Plinko's going to be on.
It's one of my favorite shows. If I could still watch it, I would.
I had an existential crisis last year that my daughter was giving a keynote speak at a governor's conference, but they had the Price is Right was in the studio next door. I was really torn. I was like, where do I go?
That is awesome. That is awesome. So you said you have Plinko set up somewhere in your home. Can you briefly explain to us how this game works, or how it incorporates Plinko really into understanding nanomedicine?
Yeah. So I mean, it's all both Plinko and nanoparticle delivery is all based off of a little bit of a statistical probability. So I work in pharmacokinetics, which is the area of mathematics describing how things transport in the body. So kind of utilizing that, and I'll bring in a little bit on here. So we just have kind of a little, the objective is that we want to get our medicine to the tumor. And so we have this Plinko board set up where we have different organs. So we have a liver, a tumor, and at the bottom are a bunch of other organs like lung, kidney, heart, spleen, and brain. For younger kids, they just like seeing what organs look like. That's their main thing. I also have a little explanation of what is nano, since you asked. Maybe this helps. I know a lot of particularly middle school level and down individuals really like this. If we think of ourselves on the meter scale, we can go down 3 orders of magnitude to the milliscale, and which would be like the size of a marble. So this is just a glass marble here. And if we go 3 scales of magnitude lower are the cells that make up our body. That's the microscale. And then 3 levels of magnitude lower than that are the nanoscale, and these particles that we're working with various diseases. So I don't know how, if you guys are familiar with Plinko or how much in your audience are familiar with Plinko, but essentially it's where this just kind of bounce along pegs and come out to like a probability of where they are. So we make different particles. We have
great. Let's do this.
We have some micelles.
Hey, before you start, what's the prize? Is there a prize that we can win?
Oh yeah. How about I owe you a round of beers when we meet up next?
Perfect. That sounds like a deal.
So we have here a syringe indicating that we can give it by an injection, or we can take it as a pill, as medicine. If we take it as a pill, an oral medicine, you can see, it got stuck. So sometimes that happens with medicines. They just kind of go into a wasteland and you don't really know where they go. We give another one by the mouth. This one is pretty common to happen, where the particle will get caught in the liver. So our liver is really specialized at clearing particles from our blood. So when we do this, a lot of them will end up getting stuck in the liver, particularly if we're giving it orally. Because as you know, Emmanuel and Pat, but like there's first pass metabolism. So if we take something orally, it then goes to the liver as a first pass and a lot of it can get removed. So
which is not a good thing, right? Because if you don't want to deliver drugs to the liver, that means there could potentially be a liver toxicity, correct?
Exactly, yeah. But if you want to deliver to the liver, it's also a great advantage too. So we have another project where we're doing that, where we are delivering to the liver, utilizing that property.
Very cool.
We can give it by injection, and we can see, okay, it doesn't get caught in the liver. That went into the lungs. We do a couple more of these. Sometimes it still goes in the liver. We have the injection and gets stuck there. It doesn't progress. That one just went out, through the kidneys, eliminated.
Josh, in terms of specificity, then, are you saying it's kind of a crapshoot? Like it's actually not very specific?
Yeah. So this is one of the main take homes I want to get is to when I bring this out is I think. When we hear nanomedicine, there's a lot of interest, as there should be. I think there's a lot of problems that will be solved with nanomedicine. We're working in the area for that.
Yeah.
But it's not a given that it's going to solve that solution, particularly when it comes to targeting.
Okay.
And I think this kind of shows that point. And I like using it in the college courses because those are the students, even graduate students, who are really hyped up on nano. And they're like, well, I'm going to put this antibody on my particle, and it's 90% of it's going to go to the tumor. When reality is, we're lucky if we can get 1 or 2% to go there.
Right. Right.
So it kind of is a way of illustrating that, and you can see with this, it's really hard to get it to go to that tumor when we go there.
Well, I think my take-home message from this is Plinko is a rigged game and Price is Right. You can tell me exactly where it's going to end up. So am I wrong to assume that Plinko is rigged?
We rigged this a little bit differently than Plinko, but it is still a statistical probability. We did some things to make it get stuck in the liver more frequently.
This is amazing, Josh. I mean, you should definitely be traveling school to school to explain nanomedicine this way. I mean, this is when someone sees this is amazing. Yeah, this is super interesting.
I think you may have to pay just a little bit of a licensing fee, but that's.
We gotta change the name Plinko to something else.
Think with 2Ks that your problems are solved.
Yeah, yeah, 2Ks.
I guess you guys, if you publish this podcast, you can take care of all of that.
Shut down before we can go anywhere. You mentioned, so you're working on TB and pancreatic cancer. Is that what you said?
Yeah. Yep.
Is there a reason why you're working on pancreatic cancer as well? Does it employ the same type of delivery system, or what?
Yeah, that's a great question, and I'm glad you linked it like that, because a lot of people think those are two different areas. And actually, it started out, I had a strong interest in pancreatic cancer. Something, so I think recently people have become much more aware of how terrible pancreatic cancer is as a disease, and that really the prognosis is very, very poor. But one of the main problems and why that is that therapeutics can't get into these very dense tumors. So in pancreatic cancer, you have these scar-like formations, this really dense extracellular matrix. And so drugs can't get into there very easily to treat the cancer cells. And so initially we were working on looking at nanoparticle approaches to overcome this barrier. And when we were in the process of doing that, we also learned a little bit about tuberculosis and how in the lung it forms these granulomas, which are very similar in structure with these dense fibrotic tissues. And there's antibiotic resistance forming as a result of that. There's some different mechanisms at play than in pancreatic cancer, but from a biophysical standpoint, which I was trained as a biophysicist, it's very similar and has the same type of barriers to go through. So we started approaching tuberculosis in parallel to our pancreatic cancer research.
Don't you have like a pet project you're working on? Do you want to explain that?
Yeah. Yeah, so actually we have a funded project through Wukini Initiative, which is a native cultures program where we are looking at how American Indian storytelling practices have evolved and the techniques that are utilized within those practices and how those can interrelate with scientific communication. And so we're looking at ways to improve scientific communication by utilizing some of these fundamental practices that have been well honed and really help for remembering difficult concepts in the natural world for centuries being passed from one individual to another. And so that's Oh no, it's just it's a fun project where I'm getting to utilize a little bit of my history training as well as the science training, particularly on the communication side of sciences. But yeah, it's a lot of fun.
I think it's really interesting, particularly that culture. It's a very how would you say, like kind of a vocal culture where they pass down stories and lessons vocally versus putting it down on paper. So that's like that's an amazing way to use that culture to teach essentially science. And I think you're pretty much, I guess, trying to do what we're trying to do on this podcast is I guess disseminate the message in a relatable way. So are you basically learning about their culture and seeing how you can kind of use science to to use the type of symbolisms or analogies that they use?
Yeah, well, so I'm learning a lot about their cultures through this and practices and I guess beliefs or setup structures as to how they approach a problem or how they may have a worldview and where that worldview comes from. We're still kind of at an early stage at this, so we're interacting with a lot of elders, Native American elders, as well as dancers, photographers, people who are doing different types of storytelling and listening to the practices that they use to remember stories that they were told from their grandparents, and how those are passed on, and looking at that technique and then finding a few threads. So I don't want to give away too much thunder because we're still in the midst of this project, but finding a few threads where some of those techniques can be very helpful to conveying the message, as you said, Pat. The objective of this podcast, right? So that we can communicate more effectively to a broader audience and have that be a memorable communication, something that 2 weeks from now that they'll remember. So
yeah, and then we'll pass it on to their kids, and their kids will pass it on.
Right. Yeah. So everybody's going to be playing Plinko Nanomedicine.
Yeah, right. That's a great marketing plan, man.
You're gonna have to trademark your game soon, Josh.
So where's your research at? I mean, are you just kind of like scratching the surface, you think, of the potential of nanoparticles in terms of tuberculosis and pancreatic cancer, or where are we at right now in terms of the science?
I think I'm at varying degrees, and it's hard to be overly optimistic. I think we all have some hopes in the field, some technologies that we have that we feel might be close to commercialization. One aspect of our tuberculosis project is a translatable aspect in which we want to, at the end of the grant, be able to start looking at this in human patients. So 5 years from now, hopefully this is something that we can think of as a realistic therapy. And that's probably the closest we are in terms of the two projects I discussed. I think there's also some interesting things that we're doing, utilizing really kind of simple concepts and unique applications. So like in the Plinko board, you saw the particles go to the liver. So we are looking at utilizing that, and we have a company we're developing now, or starting in the early stages of developing, utilizing that as a way to either prevent or treat any hepatotoxicities that happen when you're taking various drugs. So this would be things that you could take in association, say, with an anti-cancer therapeutic that may allow you to go to higher doses. It'll reduce the toxicity and be more effective against the tumor, but by doing that. So that's just utilizing more kind of simple concepts and understanding that these things that we might find frustrating because of how the body handles something, and instead go with it. Maybe there's an advantage to going with the flow, right?
So, because it seems like the challenge you have is not actually coming up with the particle that mimics, a bacteria or whatnot. It's actually making sure it gets to the right place, right, in the body. So, what's the thinking now? Like how to do that? I don't know if you heard of CAR T or T cell therapy, right? Is pretty much what they're doing. They're taking the T cells out of the body. They're messing around with it and then making it more specific to that one disease, and then injecting it into the body so that it can go and just specifically target that disease. Like, is there anything within the nanoparticle world that you can do the same?
Yeah, I think there's very similar type process. So borrowing from the immune system, like we're doing there in CAR T therapy, and their adaptability to be very specific. There are groups that have used antibodies or antibody fragments and conjugated these to particles or medicines and have seen success with those. One issue that then comes into play is if you have a nanoparticle and you put an antibody on there, well, then antibody is much larger than your nanoparticle. So what's the importance of having a nanoparticle and things like that? So we have to think about those scales. But I think there's a lot of work and potential going on there. And I think broadly as a field, I feel like maybe I'm making it sound a little naive, but there is a lot of work in that area.
Because essentially, isn't this kind of a new field?
Maybe 3 decades, I would say. Is that fair to say?
Oh, really? Okay.
Yeah, so
Well, I mean, the biggest challenge I think with the whole nanomedicine field is, not everything can be, formulated into a nanomedicine to be effective per se, right? The theory is there, but the application has been lacking. So I think that's been the challenge right now with nanomedicines, right? Not all disease states have been successful in being treated with nanomedicines per se. So that kind of which is in a way where now scientists are trying to reinvent, nanomedicine and just understanding the basics of nanomedicine and how do we use it moving forward, right? Rather than making super fancy nanoparticles or nanomedicines, can we understand the basics, right? The mechanisms of action and how to turn that into hopefully a therapy, right? In the future and stuff, right? So.
Sometimes things like this heterogeneity in disease comes out because something isn't working. We expect something to work and have a high affinity for this tissue, and when it works in one patient and not another, it highlights that there's a difference between those patients. And even when a therapy isn't necessarily working right away, it does still move the science forward in showing that, okay, there's a lot of, so this is an example in cancer where there's a lot of heterogeneity in various tumors, and now it's been identified, some of that heterogeneity has been identified by further as a result of this, and now we can still move it forward even when it's not working, but it's incremental steps.
For sure.
Yeah, that's a really good point.
Yeah. Hey, Josh, I remember from a previous conversation we had together, we've known each other for several years. I believe you've worked on something related to tattoos, if I'm correct. Yeah, I want to learn more. What was it about tattoos? There's some science behind them that you were working on, right?
Yeah, so that's funny you brought that up. I haven't thought about those in a long time, but yeah, this my PhD thesis was funded by a company developing new tattoos. So a large portion of my work as a grad student was developing tattoos. So I didn't tell my mom about that part, but that's all right.
Oh, hey mom, now you know. Now what she really wants to know is, do you have any tattoos yourself? And so how do you create a PhD surrounding tattoos? I'm kind of curious.
Yeah, so this is really cool, and I think so it's all patented and out there, so it's fun to speak about. But we're developing tattoos which could be permanent but removable, and the idea there is there's a lot of regrets in the tattoo industry, right? Somebody gets a misspelled tattoo, or they fall in love with someone and get a tattoo and then don't stay with that person. So there's a big market for removal. I think it's something like 1 in 5 individuals who get a tattoo either say they regret or attempt to have it removed in some way or covered with another tattoo or something. But the removal process is actually quite terrible. It just destroys you're basically burning the dermis layer where the tattoo is, to try and break it up to cause an immune reaction so that the cells of the immune system go and take the dye particles away. And they're really crude dye particles. And I can't take credit for developing this. So my PhD advisor, Edith Mathiowicz at Brown University, had a really great idea where she was taking water-soluble dyes and encapsulating them within non-degradable polymers. So we were making these beads that you could tattoo just like the dye and would give you a tattoo. But the one thing that we were doing with that is you could do a couple different things. You could make it a resorbable polymer such that over time that polymer degrades and that water-soluble dye just dissolves and leaves away. So you have a fading tattoo that isn't permanent. We also did where we would put in something like a small metal ion in these particles as well. So you could hit it with a low level laser that would rupture these particles, allowing the water-soluble dye to be freed and excreted. And there you have a reduced toxicity because we don't have these big clunky dyes. You're able to have it permanent. That polymer is not going anywhere, so it would stay there and could be there for the whole lifetime. But if somebody wanted to have it removed, then they could easily have it removed.
What's happening with the technology now? Is it being utilized right now in tattoo parlors, or?
I believe so. Yes, I know there was a lot of hype going back. So I think in 2007 it was the #1 Yahoo story in the nation. So it was a pretty big story back then, and it picked up steam in a couple markets. So New York, Miami, and LA tattoo markets. It picked up steam there, and surprisingly, not really for the applications that we were intending for general tattoos, but more for cosmetic permanent makeup. So, which is something I didn't realize, but, so that, generally women want to have makeup, but the process of doing that every morning or something can be painstaking. So they will get a tattoo for a permanent kind of coloration. But if they didn't want to have the same color the entire time, then this might be a good alternative. So it seemed like that was where it was picking up. It is, with the manufacturing involved and so forth, and the price going into it, the price point is much higher. So that's probably the major limiting factor of it. As far as I know, the company that does it, they changed their name a few times. I'm trying to think what they currently are. They were Freedom 2, and then they changed. I can't remember what it is. Infinite Ink was the product, so you could probably look up Infinite Ink and see where that product is. But as far as I know, it's still viable. There was also interest for military applications, so you could have for soldiers, they have identification, but that could be removed after their service is done. So yeah, I think there's a lot of applications for this.
Hey, I've always been wanting to get a tattoo, but like you said, I probably regret it a couple days later. But I mean, if you stand by your science, do you stand by your science? If you stand by the science that you worked on, are you willing to get a not so serious science podcast tattoos all over your chest and show us that it's removed the next day?
Oh, that's a big ask, but maybe not next day. I need some recovery time.
All right. So
there would be a big price tag with that, but sure, I'd do it. You guys have the price.
I mean, you definitely kind of demonstrated how you've delved into a variety of different projects, right? I mean, from tattoos to science communication, right, to nanomedicine. I mean, it's very interesting, very interesting. I mean, you have a lot of experience. I mean, this is great. Any students that's training under you, I mean, they're very fortunate to get that knowledge for sure. So are you recruiting any trainees at the moment?
This summer, I was recruiting a new graduate student and new postdoc, and I think I've identified those individuals who will be coming in now. So not currently recruiting anymore, but hopefully some of this leads to more grants, and then we can grow the lab with more students.
Hopefully through this podcast, you'll be able to attract and recruit some potential trainees as well. I mean, all the trainees that are potentially interested, they can find you on your website, right? On South Dakota State University. I believe you have social media accounts as well.
Yeah, so at Reineke_lab is my Twitter handle. So yeah, thanks for the plug, Emmanuel. That's always helpful.
Yeah, have you always had that passion for science, Josh? Like early on, and
I think for me, biology growing up was an area that I just had a lot of curiosity. I think that's how a lot of people start. When I was an undergraduate, my very first year, my freshman year, I was pretty nervous, but I went and approached faculty and looked if there were any research positions available. And most of them were, we need somebody to wash dishes. But I was really lucky.
Yeah. Karma, are you now looking for undergrads to wash dishes?
No, in fact, I say, because I was so fortunate, I spoke to Edith Mathiowicz at that time, and she matched me up with a really great graduate student, Chris Thanos, and he really helped develop individual project for me and really fostered that passion within me for the field. And it's really, to the two of them, is the reason I'm in the field that I am, because I just really enjoyed that experience even early on. And so now when I have undergraduates come in, I do something that's a little bit unconventional, is I have them do a shadowing. So I have them shadow the students and postdocs in the lab for 2 weeks before there's any commitment, before I make commitment to them, before they make a commitment to doing work in the lab. Because I want them to really understand it. I think it's so complex outside to say, okay, what is research, and get that idea. And then from there, I have a conversation with them. What is it you really want to do? How can we meet like your life ambitions? And you may not know at this stage, but at least they'll have some things like, oh, I saw this, and I really thought this assay was really cool, and I want to pursue that more. So we'll put them to work on that assay and become an expert in that assay. Or they may develop into independent projects because they really, because a loved one had pancreatic cancer, they just really want to work on that pancreatic cancer project, and they don't care about what aspect of it, but they want to help move that forward. And it's taking the time to really listen and see what it is that they want. Sometimes, I had students say, "Yeah, that's not for me," and walk away. And I say, no hard feelings either. It's important that you find what you want. And I think I was fortunate to have that type of experience with Edith, and so I try to share that with students as well.
I think it's important. It's unfortunate that not many jobs or careers offer this kind of shadowing or volunteer opportunity. Because it's very challenging as a young individual, right? When you're trying to choose a career path, you don't know what you like until you try it. Right. So it's unfortunate that not a lot of jobs will give you that opportunity. So, but, with you being able to kind of allow students or potential graduate students to kind of just watch, right? I mean, just watching the different projects, kind of learning the challenges of research, understanding the time commitment, towards reading literature and problem solving and so forth. I think it's a great idea, definitely a great idea, and really help that student understand is research what they enjoy, or are they looking for something else? Because, when people hear research, I remember hearing research for the first time, I thought it sounded super exciting. I was like, wow, I want to save the world. Once, my first research opportunity will allow me that potential to save the world. But unfortunately, that's not the case, right? There's a lot more to just saying that you can save the world, right? There's a lot more work that is needed for sure.
Yeah, I think it's really important. What you're doing is like you're actually vetting like for that passion, right? Because I mean, a lot of people know. Being research in a lab, going in on weekends to keep your cells alive, or it's not all rainbows and unicorn farts. It's like a lot of hard work and not a lot of money. So that passion is really important. And I can speak from experience. I was on my way to do a master's degree too, and I within 2 days in the lab, I was like, that's not for me. So I actually went like the corporate route. So good on you for that. Because actually, that shadowing thing I think it's huge. What Emmanuel was saying, for the reasons he was saying, I don't think not enough people actually do that. So
yeah.
Good for you.
I was just going to echo exactly what you said, Pat. I usually kind of flip it on its head and say that science usually doesn't work. That's just the fact of the matter. Is most of the time the experiments don't work. But it's interesting if it's something that you're really passionate about. Suddenly, when it's not working, it becomes, I want to know why that's not working. That's really curious, and it really becomes a driving factor. Whereas if it's something that you're not so passionate about, then it's just kind of you're running into a wall. Like, I can't stand this. Nothing works. It's terrible. It really because it isn't going to work. I tell my students, it isn't going to work, but be happy when it does, because usually it doesn't. So expect that.
So coming back to your research, what are your plans like, for the future? Congratulations on this NIH grant. That's amazing. And how long is that lasting? And obviously you're constantly applying for grants, but where do you see your research going?
So we have that grant is a 5-year grant, and as I stated at the end of that, we hope to do some translational work from that, assuming we hit all the milestones. So I really hope that's something that continues. Whenever you're working in an area, there seems like there's so many avenues you can pursue. You kind of have to shut them down sometimes. But one of them came up recently that I didn't realize when working with this tuberculosis, was that I'm in an agricultural school, and tuberculosis is actually a very costly disease in dairy cattle, as well as in buffalo for meat production. And so we just got a small grant to look a little bit at that. So if you would ask me a month ago, where do you see yourself going? It might be a lot different than right now. There's, this new potential of we might be looking in kind of that agricultural space and where it can go.
So currently at this point, there are no vaccines for tuberculosis for animals or humans at all?
No vaccines. Treatment in humans, there's lots of antibiotics, and usually it's a combination of those antibiotics that's used, and it's successful in many cases if the patient can follow that long-term protocol, which is very difficult. So as a result, there's been multi-drug resistant forms that have developed. So that's one problem in human disease. The other problem in human disease is tuberculosis is a very slow-growing bacteria. And so a lot of the reason you haven't heard about a lot of individuals being infected with tuberculosis because they will never develop the disease from it. But if we want to think about eradicating it, we have to be able to treat this latent bacteria. And the bacteria that we, the current treatments just don't treat the latent bacteria. A lot of antibiotics are dependent on the rapid division of the microbes, and this is very slow dividing microbes. So there's that challenge. So really there's a lot of barriers to that treatment there.
So what is multi-drug resistant? You mentioned that. How does that play a role?
It just, so a lot of the frontline therapeutics, like rifampicin is one, the bacteria are resistant to it. So you can treat, even if the drug does get to that space, the bacteria will continue growing. So there were rifampicin is resistant tuberculosis. So then therapeutics then evolved to use multiple antibiotics, and now there's drugs which are microbes which are multi-drug resistant, so they're resistant to 3 or more of the antibiotics used in tuberculosis. And now we have also extra drug resistance, XDR tuberculosis, which is resistant to 5 or more of the drugs and is largely untreatable.
So you're talking about resistance. So does that mean that the tuberculosis, bacteria is actually changing, right? It's mutating?
Correct. Yeah. So that it develops a mechanism such that drug is no longer effective against it. So.
Right. So in terms of coming out with nanoparticles, I mean, is it through that you constantly have to change the nanoparticle as you're over the next 5 years? Potentially it could be different.
I think. So usually we think of nanoparticles as a mechanism to bring a drug there. So we can maybe prevent some resistance, because some of the resistance might occur by not having enough drug in that space next to the bacteria. So if you give the bacteria just a small amount of drug that isn't lethal, it will allow it to develop these mechanisms to resist that drug. So that's probably the biggest area nanoparticles can work. But there's others who have worked with, say, silver nanoparticles as a way where the nanoparticle itself is a therapeutic. It disrupts the membrane of the bacteria. But there's been some interesting studies, and I don't know fully because I don't really work in that area, about how there has been some resistance developed even to that. And so when you start thinking about that, then I think the point you made, Pat, is exactly true. You have to keep evolving then, just as we are evolving drugs, we have to keep evolving our nanoparticle systems to address the new barriers that come up. We're trying to hit it at a higher level and not actually treat the bacteria itself. So the macrophages are the cells that house these bacteria. They're very efficient at clearing infections in the body normally, but the bacteria is shutting it down in this case. So we are just trying to interrupt that process so that the macrophage regains its ability to clear it. So utilizing the natural defenses that already exist and really help support the cell in doing that process. That way we don't have to worry about any changes in the bacteria, because we're not trying to hit the bacteria. But we might have to worry about changes in the cell. So
that was interesting. So it's basically a different approach. Instead of targeting the bug in our body, you're trying to basically prime our own body or make our bodies recognize the bug itself. Pretty much.
Yeah. So the field is called host-directed therapy. So in this case, the host is the cell. So we are directing our therapy to that host to then help it eradicate the infection.
Any potential side effects you think will arise from this? Because like you said, these macrophages or these immune cells are important in our body. Will modifying any host cells potentially result in higher susceptibility to other infections potentially?
I don't know about susceptibility so much. I think though it is definitely a concern and something we have to look at. Is there is the possibility to overactivate your immune cells, right? So if you have that overactivation, then you can have a lot of different problems. So I think this is something in cancer therapy that's been seen with some of the CAR T cell therapies and such, where you get such an effective killing, you get a cytokine storm, where all these cytokines and debris are released that activate the immune system, and it really can overwhelm the immune system and cause its own issues and potentially even death in the patient. So we have to worry about the similar type thing happening. We can't have a very rapid type therapy. And so there's going to be some dosing that's going to be needed. And we don't yet understand that. And that's hopefully things in our research that will elucidate a little bit. But certainly there's always the potential for adverse reactions.
I mean, the term cytokine storm it's been popping up a lot in media, especially with COVID, right? So that's kind of interesting how cytokine storms seem to play a role in a lot of different disease states, and there's still a lot that we need to understand about cytokines and how they protect our bodies as well as cause disease in our bodies. So it's very, very interesting.
And speaking of COVID, just curious, what do you guys think about Russia coming out with the first vaccine?
Well, I was going to say that when Emmanuel was saying it's going to take a while and everybody's waiting for a vaccine, I was going to say, what do you mean? It's already here.
I should rephrase. I'm waiting for an effective vaccine. That's hopefully 99.9% effective. I mean, you could come up with a vaccine, but if it's only 20% effective, right? That may be a different story, right? It's kind of like the flu vaccine, right? We get the seasonal flu vaccine. It's not 100% effective. It's got a low effective rate, but yet it's better than nothing, right? So
is your lab still closed then, Josh?
No, it's open. It's kind of in a phased open, and we're learning how to do things new ways too. So I have like a senior graduate student. Or I have a junior graduate student who needs to learn cell culture technique, and when they can't be right with somebody, how do we do that? So I have a senior graduate student who's using a GoPro to talk through as she does the assays and really highlight the important things. He will then view that, the junior graduate student will view that and then ask questions and then make a attempt of doing it while he's recording it, so that then, we can jump in and critique that video like, oh, you forgot to do this. Yeah, I think it sounds terrible, right? But we have to think of new ways to do things and still, still put out quality science, still meet the objectives of training new scientists and still develop our science.
These are excellent examples that you've given where we have tried to learn and adapt. Maybe it might even be a permanent training method that you're going to implement later on with your research team, even when there's no COVID.
I think, but the chair of my department heard that the potential of what we were doing, and he's like, "Oh, do you mind if we share those videos?" I don't think my graduate student was too keen on that. Like, no, I don't want to.
You see, I told you. I mean, you're doing something great. I mean, other people want to be able to learn from you as well, for sure. I mean, hey, I want to take a look at these videos and maybe I can implement them in my research team.
You're gonna scare all my graduate students away. They're never gonna let me see the videos now.
Excellent. Yeah, thank you so much, Josh. This was an amazing interview with you. I had the pleasure of learning all about your research program. Some of the hobbies that you've been doing to kind of just maintain sanity as well with everything happening with COVID and so forth. But yeah, it was a great pleasure. And just so everyone knows, your website can be searched on the internet, right? South Dakota State University. You have your own website there. So if anyone that's interested in potentially pursuing graduate studies under your supervision, they can just contact you directly. Your email is there, and as well as social media. So definitely go ahead and follow Josh on social media. He's definitely a very smart and fun and really approachable individual. So anyone that's interested in learning from Josh, I say go ahead and send him an email.
It's really refreshing to see such a well-rounded person like yourself and like Jill on the last podcast. That's you both have been just breaking down the stereotypes of what it means to be a scientist.
Well, that's good. Maybe we're developing new stereotypes, but that's good.
Hey, again, we're adapting just like bacteria, right? We gotta evolve and adapt as well. Science is the new cool now. Thank you very much, Josh. Take care and enjoy the rest of your week. See you later. Bye-bye.